예, 안녕하세요. 감사합니다. 우리 학술 웨비나에 이렇게 참석하게 되어서 정말 영광입니다. 어, 제가 한국어로 과학 발표를 하는 게 익숙치 않아서 영어로 준비하게 된 점 양해 부탁드리겠습니다. So today I'll talk about three topics. That is histone modification, protein engineering, and LSD1, which stands for lysine specific demethylase 1. And it's all in the theme of epigenetics. So I'm interested in understanding how histone modifications help control gene expression. So I'll start explain a little bit about histones, which is a part of a nucleosome. So nucleosome, which is a fundamental subunit of our chromatin in our nucleus, is made of four different types of histone proteins. Those core histone proteins are made of histones H2A, H2B, H3, and H4. And there are two copies of each histone molecule in the nucleosome. So therefore, there are 8 histone molecules forming an octameric protein complex. This octameric protein complex is wrapped around by about 150 base pairs of DNA, making a nucleosome unit. So when we think of nucleosome, typically when we learn from the textbooks, we typically think of DNA compaction or chromatin compaction implicated in like gene repression or implicated in DNA replication. But in reality, these nucleosomes can directly participate in helping control gene expression. And oftentimes, during that process of gene expression controlling, this histone N-terminal or C-terminal tails that are flexible are often chemically modified. So today, I'll focus on one of the chemical modification called methylation of lysine, specifically on one type of histone protein called histone H3. So this histone H3, lysine 4, we call H3K4, can get methylated by KMT2 family methyltransferases, and it can go from lysine monomethylation all the way to dimethyl and trimethylation. And what do these modifications do? So it turns out that when lysine 4 gets methylated by this methyltransferases, these methylated lysines can help recruit various proteins. Some of them are chromatin remodelers, transcription factors, transcription regulators, or RNA pull-2 complex itself. So a lot of these proteins recognizes methyl lysine that help promote turning on the genes. So they are implicated in gene activation. Cells also have dedicated erasers for this methyl mark because when cells finish expressing genes, they want to turn those genes off. One of the main lysine demethylase that can erase this methyl mark from lysine 4 is LSD1, lysine specific demethylase 1. In the active side of LSD1, there's a flavin cofactor that helps the removal of methyl moiety from the lysine through the redox chemistry. And this LSD1 has a binding partner protein called CoREST that help LSD1 recognize a nucleosome and stabilize LSD1 molecule. So inside of our cells, there's a constant tug of war between methyltransferases and LSD1. So methyltransferases help install methylation groups on the lysine 4 that turn on the genes. And when cells finish expression, they remove these methyl groups with the help of LSD1 demethylase. So this really help control the genes really tightly. So more than half of our histone proteins in our cells are actually multiply modified. So meaning that it's not just likely to have methyl lysine 4, but it can also have other types of modifications simultaneously on the same histone protein. So recent mass spec experiments pointed out that lysine 4 often gets co-modified with lysine 14 acetylation. So this lysine 14 is 10 amino acids away from lysine 4, and acetylation of lysine typically is implicated in turning on the genes. And there are actually quite a few interesting facts about this lysine 14 modification. So when there's a lysine 14 acetylation, it promotes the activity of KMT2 family methyltransferases. So it can almost more quickly, efficiently methylate lysine 4 of histone H3, which would turn on the gene more strongly. On the other hand, if you have this lysine 14 acetylation, people in biochemistry field have found that LSD1 becomes extremely slow in demethylating lysine 4. So it's kind of as if this lysine 14 acetylation is protecting methylated lysine 4 against LSD1. But this was all shown biochemically, and there have not been a lot of cellular data to support this idea of this crosstalk. So we call this crosstalk between the two marks, chemical moieties. And simply, we thought that this acetylation of lysine 14 will be implicated in enhancing the gene expression. And this kind of makes sense. So I was thinking, what kind of if it's implicated in gene expression, what kind of biological roles would it play? What kind of system? So this is one of the examples I try to come up with to sort of conceptualize our biological questions. So for example, there's a phenomenon called epithelial to mesenchymal transition, where a lot of epithelial cells, in response to stimuli such as TGF-beta, it undergoes these morphological changes. It undergoes a lot of morphological change while undergoing significant transcriptional changes. And LSD1 is thought to play a key role in this process because in order for EMT, epithelial to mesenchymal transition, upregulated LSD1 is thought to turn off the genes that are linked to epithelial phenotype that facilitates acquiring mesenchymal phenotype. So this is how we envision it happens. So this is a genomic region where a lot of open chromatin promotes a lot of gene expression. And imagine this: a lot of these genes in this chromatin is linked with epithelial phenotype. And during this EMT, LSD1 will come in and remove this methyl mark, which is colored in, which is this red triangle, that will silence them, and therefore it can turn off the epithelial phenotype. Imagine some nucleosomes are decorated with acetylation in yellow circle and methylation in red triangle. Then LSD1 will be really, really slow in demethylating these genes because it cannot demethylate well. And therefore, it would allow some genes to be resistant to LSD1 while this EMT is happening, because EMT is thought to be systematic. So it is thought that LSD1, they don't just randomly go to this open chromatin and just randomly demethylate genes for gene repression. It is thought that this event is happening systematically. And we think this acetylation is a key histone modification that allows LSD1 where to go or where not to go. And on top of that, LSD1 actually does not have any DNA specificity. So we think this acetylation of lysine 14 that is co-modified with lysine 4 methylation is serving as a navigator of LSD1. So the thing is, how can we demonstrate this in cell? And so we thought one way to address this kind of interesting biological question is through protein engineering. So on the left, imagine you have wild-type LSD1 recognizing the nucleosome that is positioned at either promoter or enhancer regions of certain genes. And you have this nucleosome contains methylated lysine 4, acetylated lysine 14 that would resist LSD1 to demethylate the histone. Therefore, it's continuously turning on the genes that allows these genes to be on while other genes are turned off by LSD1. Therefore, this systematic gene repression could be allowed. On the other hand, if you want to identify what these genes are that are protected by acetylation, we thought this protein engineering approach could be viable. For example, LSD1 mutant that is no longer caring about acetylation of lysine 14 would rapidly demethylate these histone proteins that will turn the genes off. So, for example, this specific genomic loci, which is decorated with acetylation of lysine 14 and methylation of lysine 4, will immediately get removed by LSD1. And therefore, if you compare the two cells, one cell just expressing wild type LSD1 versus the other cell expressing mutant LSD1, we can do comparative analysis to identify what are which genes or which genomic loci can get are presumably decorated with methylysine and acetylated lysine where the crosstalk is occurring. So I hope we're on the same page here, because this justification of protein engineering sometimes is the one that the part that confuses people. So I would be happy to address your questions later on, but this was our idea. So to do this enzyme engineering, there were several things that we're considering. So first of all, we are thinking about mutating active site of LSD1 to modulate this LSD1's sensitivity toward lysine 14 acetylation. However, when you change the active site, oftentimes it can really greatly perturb the activity or other properties of protein. So we really wanted to minimize the changes within the active site while acquiring that losing the sensitivity toward lysine 14 acetylation. So that was our number one goal. Second goal was that when we mutate this enzyme, we did not want to greatly change the activity toward histone substrate that is just methylated on the lysine 4. So meaning that even if you make the enzyme faster, if you globally make the enzyme faster, then we cannot really compare apple to apple, orange to orange. So we wanted to maintain the activity toward unacetylated histone tail almost the same compared to the wild type. Third, we wanted to selectively enhance the activity of this demethylation when the substrate is acetylated at lysine 14. And lastly, oftentimes when you mutate proteins, it can undergo instability. So we wanted to maintain the similar cellular stability compared to the wild type. So those were our 4 goals and 4 considerations when engineering this enzyme. And to do that, we wanted to gain some insights from structural studies to pinpoint which residue to mutate. So luckily, there was a crystal structure that was published in 2007 from Dr. Andrea Mattevi in Italy. So this is a crystal structure of LSD1 in green, bound to co-rest in its binding partner protein in blue, and also bound to the histone substrate peptide. So if we zoom in to the active site, this is a flavin cofactor where it catalyzes demethylation for lysine 4. And this is a histone peptide in orange, and green residues are coming from LSD1. So at the catalytic site, there are 2 key contacts between this histone protein and LSD1. So lysine 14 of the substrate, histone peptide, is making an electrostatic interaction with glutamate 559 of LSD1. And also, this lysine 9 of histone peptide is making a polar contact with histidine 564 of LSD1, which also forms a hydrogen bond with threonine 6 of histone peptide. If you mutate these 2 residues to alanine, it really slows down the activity of LSD1, highlighting the importance of these 2 key interactions. So at this point, we reached out to Andrea Mattevi in Italy, and we solved the crystal structure first of LSD1 in complex with the histone substrate that is acetylated at lysine 14. And I forgot to say, this lysine 4 was mutated into methionine because methionine can mimic methyl lysine. So we solved the crystal structure of the same system, but bound to acetylated state of lysine 14. And those two structures are superimposed here. And first of all, we did not see much structural changes overall. Their backbone topology is nearly the same between the acetylated and unacetylated peptide bound state of the peptide. But there's a subtle difference, but quite distinct. So when acetylated lysine 14 is bound, peptide is bound, we see that this salt bridge between lysine 14 and glutamate 559 of LSD1 is now broken because it's no longer a positive charge here. What that did was it pulled this lysine 9, which was facing down originally to make a polar contact with histidine. It now pulls up that lysine and makes an electrostatic contact with glutamate 559. And what that did was it made this histidine kind of all alone or just bonded to threonine 6. And we don't know how this translates to catalysis exactly, but what we rationalized in this case was that this lysine 9 movement upward due to the salt bridge breaking of the salt bridge here was the key in slowing down the enzyme. So we thought maybe we should mutate LSD1 where we can regain this polar interaction between lysine 9 and histidine 564. So that was our rationalization when we solved this structure. So for the first 2 years of my postdoc, I was continuously mutating a lot of residues around the active site of LSD1. And after mutating about 30, maybe 35 residues, we finally found one mutant that does the work. And it's actually pretty cool because the mutation was not exactly at the catalytic site. So I would like to highlight this residue, tyrosine 391 of LSD1. This tyrosine 391 is not at the nearby the flavin. It's about more than 30 angstroms away from flavin. However, it's at the CoREST1 binding interface, which is a binding partner of LSD1. So we mutated this tyrosine into a lysine. And the reason was, you see, when the substrate has acetylation, which neutralizes positive charge on the lysine, it loses positive charge, and therefore we wanted to introduce the positive charge to regain the balance of the net charge. And because of this second shell effect, we intentionally chose a residue that was a little away from the active site to minimize the perturbation of the structure on the active site. And this did the work. So when we mutated tyrosine right at the binding interface between CoREST and LSD1 into a lysine residue, we crystallographically see that this lysine 9 is now moving downward to nearby this histidine 564. So at this point, we're pretty happy to see this result, and we're puzzled, how does this actually happen? One of our rationale is that this part of CoREST has actually a lot of basic residues, lysines and arginines. And because we introduce a lysine here, it can causes repulsion, allowing this CoREST to move down again, downward. And this charge cluster is somehow causing the long-range coulombic interaction between lysine and arginine and lysine residues. And therefore, it's favoring this slight orientational change of lysine on the histone. So our biochemical data was consistent with what we observed with the structure. So when we mutated this enzyme and did a demethylation assay on the nucleosome substrate that has both acetylation of 14 and methylation on lysine 4, now we can rapidly demethylate lysine 4 opposed to the wild type enzyme, which was extremely slow in demethylating the substrate. So, which was pretty cool. And even cooler, when the substrate had a methylation and acetylation, this mutant's activity was almost comparable to the wild type's activity toward unacetylated substrate. So this one was almost matching up with our initial kind of a goal, what we wanted. And so at this point, we are pretty confident about, biochemically confident about this enzyme is capable of demethylating the histone nucleosome substrate, even when the substrate has acetylation on top of methylation. So how does this biologically play a role? So to answer that question, so after the first 2 years of biochemistry, the next 2 years of my postdoc was I focused on cell biology. So in collaboration with Dr. Brian Lau at Harvard University, we did a CRISPR knock-in and try to characterize cellularly what's going on to understand what's the biological function of this crosstalk between acetylated histone and acetylated and methylated histone. So we did CRISPR knock-in. We could not get homozygously edited cell, first of all. This K562 leukemia cell, chronic myelogenous leukemia cell, it's a triploid cell, so it has 3 chromosomes. I tried really hard, but I could only get 2 out of 3 chromosomes mutated into Y2K tyrosine to lysine mutation. But because this mutation is not causing loss of function, but is causing gain of function selectively toward acetylated substrate. So we thought this heterozygous editing was sufficient to do comparative analysis between the unedited versus edited cells. So first thing we checked was that when the cells were edited, it had similar LSD1 expression level between the edited versus unedited, which was what we wanted. Second, their growth rate was about the same. So there wasn't really outstanding phenotypic difference between the two. But it also meant maybe perhaps LSD1 was the basic function of mutant LSD1 was probably not very different from the wild type, which was fine with us. And it had the overall cells had similar drug sensitivity between the two. For example, Gleevec was pretty potent in killing these cells. Where we actually saw the difference between these two cells were actually their transcriptomic levels, because LSD1 is implicated in turning off the genes. So this is RNA-seq volcano plot. And the left side of this 0 indicates genes that are turned downregulated compared to the wild type that are in mutated cells. And on the right side indicates genes that are upregulated in the edited cells. Especially the ones that are in blue or green dots here are the ones that are significantly altered. And because we thought the LSD1 is a gene repressor, so it's implicated in downregulating genes, and the gain of function will allow further genes to be turned off, we focused on downregulated genes. So a lot of these genes, about 500 genes, were related to cell adhesion or morphology. Are they really turned down because of the fact that they are better demethylated at the acetylated histone? So that was our question. So we did this experiment called cut and run. I'll quickly explain what cut and run is. You add antibody to the cells, and antibodies will recognize methylated lysines on the in the nucleus. And you deliver this micrococcal nuclease that recognize this antibody, and it can cut the DNA around it. We get the DNA out, and we do next-gen sequencing. And the output, where the y-axis is a signal, and the x-axis is a genomic loci, genomic coordinates. These peaks indicate the enrichment of those nucleosomes containing, for example, methylated lysine. And since LSD1 mutant has a gain of function, it's implicated in removing those methylated lysine better than the wild type. And indeed, that is what we see here. So a lot of these downregulated genes had lower levels of lysine-4 monomethylation level compared to the edited cells. And on top of that, a lot of these genes had higher level of stronger enrichment of lysine-14 acetylation compared to unaffected genes. So at this point, we are pretty convinced that our biochemical expectation from this mutant LSD1 was matching up with LSD1's cellular function, where LSD1 mutant can go to genomic loci, that are heavily enriched with lysine 14 acetylation and is capable of demethylating this histone better than the wild type, causing gene repression. So basically, in our summary, we created this Y2K mutant LSD1 that is capable of removing the methyl groups on lysine 4 of histone when the histones are co-modified with lysine 14 acetylation. And we did that just to understand the biological function of this crosstalk between lysine 14 acetylation and methylation of lysine 4 on the same histone protein. And to what we found is that a lot of these genes that are presumably co-modified are related to cell adhesion and morphology. And cut and run data indeed indicate this crosstalk between these two marks. The thing is, initially, I mentioned about EMT as a model system. Unfortunately, K562 cells do not undergo EMT, and they're actually really poor at, they have poor adhesion to other cells. So for the future study, hopefully in my own lab, I hope to study this EMT phenomena that are regulated by this crosstalk between methylation of lysine 4 and acetylation of lysine 14. And interestingly, a lot of this crosstalk are thought to be also implicated in other types of cells, such as endothelial cells, or also in case of pathogen-host interaction. So I want to explore further histone crosstalk implicated in different biological systems. One thing for the remaining time, about 5 minutes, I also want to share some unpublished data because what I learned from this, my about 5 years of this postdoctoral research to study this crosstalk was that we actually need a better tool. And I'll try to explain what I mean by that, and then I'll share some unpublished data. So when we look at our cut and run result, this is how we try to deduce the crosstalk between methylation and acetylation of lysine 14. So for example, you see like ChIP-seq or cut and run, you see peak here. Okay, so this peak indicates there are some methylated lysine 4 at this specific genomic loci in the form of nucleosome. And you also see a peak here at the same loci co-occupied with lysine 14 acetylation. And you think that, okay, so because you see the enrichment at the same genomic loci between methylated lysine and lysine 14 acetylation, it's likely that the nucleosome have this methyl and acetylation mark on the same nucleosome at the same time. In reality, we are not really sure about that. And I'll try to explain why. And there are different, of course, different patterns of modification. In the beginning, I mentioned that nucleosomes have 2 molecules of histone H3. So there are two molecules per each core histone. And if you say that this nucleosome has methylation of lysine 4 and acetylation of lysine 14, it can actually have 2 meanings. One is that methylation and acetylation can occur in 2 different histone tails, and I call that in trans. Or this nucleosome can have methylation and acetylation on the same histone protein, which we call in cis. And if you use a conventional antibodies, which is used in ChIP-seq and cut and run, you cannot really distinguish if these modifications are occurring in cis or trans. And a lot of biochemical properties of many enzymes, including LSD1 or other enzymes, have different properties of, they experience different sensitivity, whether these modifications are in cis or trans. So we need a better tool to distinguish if they're modified in cis or trans. And this is a specific tool I want to develop. It's kind of like antibody, but it will only detect if the modifications of these PTMs on the histone proteins are co-occurring on the same polypeptide. And imagine you have this kind of tool. What you can do is you can have a ChIP-seq that you can, you can add another dimension into the data. You can detect methylation and acetylation co-occurring at specific genomic loci, and you can compare that with the cells that are treated with different condition or different diseases. So to do that, I took a little bit of the new and a little bit of conservative approach to design such a tool. What I did was I found that there are certain naturally existing domains that can recognize lysine 14 acetylation quite selectively, but slightly weakly, about KD value of about 10 micromolar. And I found some plant proteins, some domains that can recognize this chemical methyl lysine selectively, but weakly. And this is where I decided to use some cutting edge protein design tools where I can design some linker that can allow this specific conformation of the two domain in fusion to attain the avidity effect toward the target that only has methylation and acetylation. So this was the kind of the pipeline of how I designed. I chose this bromo domain and PhD finger. First, I did a docking of the two in complex with the substrate in AlphaFold. And I used the Dr. David Baker Labs RF diffusion and protein MPNN to introduce the rigid linker between the two, hoping that it attains the selectivity through the avidity effect. And indeed, that's actually what we see here. So when we did some binding affinity assay with the toward the target, peptide target that has methylation of lysine 4 and acetylation of lysine 14, it has about 0.6 micromolar binding affinity, where the control peptides have a more than 20-fold reduction in affinity. So we think there is some avidity. We can control the avidity and selectivity through this protein design mechanism. And this is although the binding affinity itself is not really strong, if you attach a GFP protein into the current prototype, you can actually use it for Western blot. So you don't have to use a primary antibody here. This protein with GFP fusion can serve as a primary antibody. And you can blot specifically histone proteins that are modified with lysine 4 dimethylation and lysine 14 acetylation. So with that, I would like to thank everyone in the collab and my collaborators, Matevi Lab, Lea Lab, Croda Lab, and Back Lab. And I would also like to thank my family, especially my wife and son, and my parents-in-law. And thank you all for listening, and I would be happy to answer your questions. Thank you.